Okay, welcome everyone joining in. Welcome to today's webinar, RNAi and Varroa mites, a new technology for managing an old pest of honeybees. My name is Laura Isles. I'm the co-director of the North Central IPM Center. We're one of 4 regional IPM centers supported by USDA NIFA Crop Protection Pest Management Regional Coordination Program. We have a mission to coordinate IPM across the region by sharing and promoting IPM research, extension, and teaching activities. If during today's webinar at any time you have questions, please type them in the Q&A part at the bottom of your Zoom screen, and our speakers will answer those questions at the end of the presentation. Again, I'm happy to welcome Dr. Matt O'Neal and Jarod Perez from Iowa State University. He's there too as well. And so I will let you all take it away and give us some information on honeybees.
Thanks, Laura. Thanks to North Central IPM Center for hosting us today. Thanks for everybody for joining on. My name is Matt O'Neal. I'm a professor here at Iowa State University with a focus on insect pests and mostly of annual crops. But over the last 10 years, my lab has expanded to look at how our landscape affects honeybee health and productivity. And most of the work has been looking at how conservation practices can improve honeybee health and productivity. But anybody who works with honeybees is well aware that maybe the number one pest, the number one problem for beekeepers year in and year out are Varroa mites. And today we're going to talk about the Varroa mite and a new technology that's now been made available for our beekeeping community. But this talk is also a bit bigger than that. It's going to address maybe even larger issue that's hinted at in this question. So what do these 3 pests have in common? And, oh, yep, there it is in the polling question. Let's take maybe 30 seconds for people to post their answers.
I can see the answers.
Maybe read a couple of them.
Yeah. So we've got no wings. They have RNAi pesticides designed to target them. A scientific name that I can't pronounce, Leptinosauria declamata. Exoskeleton
Ooh,
destroys the hive,
okay.
transmits pathogens.
I'm going to stop you there because
Yeah.
there's a bunch of good answers there. Unfortunately, there's some wrong ones. 2 of the 3 have wings. So I'll just go through. This is the western corn rootworm, this is Colorado potato beetle, and this is the Varroa mite. Varroa mites don't have wings, but they do travel on winged insects like honeybees. But they all 3, as one of the participants noted, have a new form of pesticides, RNAi, that are available to people who are trying to manage them. And what maybe even further up, what they have in common is they've all developed resistance to conventional kind of existing pesticides, which is part of the reason why RNAi, this new technology, has been investigated and explored to manage these pests. And so we'll come back to this topic, but the most recent form of this technology, RNAi, that's been developed for pest management is attacking the Varroa mite. So let's talk a little bit about that. So the Varroa mite is got a terrific name, Varroa destructor, and it's was brought to the United States inadvertently as an invasive several years ago, and has become the number one pest of beekeepers throughout North America, if not the world. And I apologize, I should have introduced Jarod Perez. He's a graduate student that works in my lab who's been working on our honeybee projects. So I brought him along to make sure I get this right. But there's 3 pictures here showing what the Varroa family looks like that could occur in the hive from the early stages of its growth all the way up to the adult mother, the red, and the adult male. And the adult female is shown here in the picture. That's not life size, but in this picture with the pupal stage of a honeybee, you can get a sense of how big this critter is. And to give you some idea, if you built this up to human size, that'd be about the size of a regular dinner plate. Honeybees typically aren't able to remove them. There are some strains, lineages of honeybees that'll groom themselves, but the mite is a terrific pest because it both feeds on the adults, but also feeds on the larvae. So here in this picture, you see the life cycle of the mite. The queen is laying an egg. That egg is being nurtured by the worker bees, who often in an infested colony move around with the mite attached to them, who then moves their off of the worker adult bee into the cell and starts feeding on the immature stage that eventually develops into a new adult. And a couple generations can occur inside that cell, and that new worker goes out and continues the life cycle in other cells. And as we pointed out, the mites lack wings, but they are able to move around inside the colony and from colony to colony through this process of phoresis. They attach to the adult workers that have wings, moving with them as they nurse the bees inside the colony. And then if they happen to move around, rob other colonies, disturb other colonies, pass along the mites in that way. And beekeepers who keep bees in dense stands or apiaries can often have the mites spread from one colony to almost all of them. Is that fair to say, Jarod?
Yes.
Thank you. And then the impact of the mite is both direct and indirect. This is a picture of a cell that has been cleared out because the brood died within it. And then here you see an adult that's emerged but has suffered the consequences of a virus infection that was transmitted by the mite.
In the photo on the left, there's actually 4 adult mites.
Oh, yeah.
They're really hard to see.
Yeah, I got to look at it. So this is routinely identified by beekeepers as their #1 pest. And it often contributes to the high rate of overwintering losses that occur in this part of the world. So what can beekeepers do about this? Well, I asked Jarod to come help me get this point across, because there's a variety of treatments that are currently available and have been used for several years now to manage the mite. These are 4 that we've used in our apiaries and many of the beekeepers around us. So amitraz, I think it's fair to say, is the most common, most popular.
Most.
Yeah, and it's a neurotoxin like many insecticides. The treatments take 6 to 8 weeks, and they're applied, as you can see here in this strip. The workers that touch the strip spread the treatment throughout the colony, and this helps kind of manage within a colony the mite load. The advantages is it's not temperature sensitive. It can be used in a variety of situations. It specifically targets the Varroa mite, so there's limited impact on the bees. The disadvantages is it does have some moderate mammalian toxicity, so you have to avoid using this when you're trying to harvest the honey. And then the biggest disadvantage is the recent occurrence of miticide resistance or amitraz resistance, which recently has been identified as contributing to some really high rates of bee die-off from one year to the next. So this leads us to think about the other treatments that are available. And I'm going to just walk through those and kind of keep in mind why amitraz, something fairly easy to apply, fairly safe to the bee, might be the #1 management tactic, and these others not so much. So formic acid, strips are added in brood nests, as you can see in the picture. Treatment takes about 14 days. It vaporizes within the colony. Some advantages, again, targets the mites inside the cap brood cells, so you can manage them before they're kind of moving around. It kills both the immatures and the adult mites. Some disadvantages are that it's temperature sensitive, easy to dissipate, something that in the heat of the summer, even though the honeybees regulate the colony, you're going to lose some efficacy. Too intense for small, weak colonies, so probably not something that we're going to be able to use when a mite load gets really high. And it requires proper ventilation, so there is some impact that could occur to the bees. in part because the vapors that are produced from those strips can interfere with the queen pheromones and prevent the worker bees from attending to her needs. And you get young brood fatalities if the vaporization has too much of an impact. Oxalic acid, it's not clear to us what the mode of action is, but appears to kill the varroa mites when they come in contact. You can see the application being done here. Multiple ways to apply, both in terms of what the drip application. These are some strips.
And there's drip, strips, and you can also vaporize it using a fancy vaporizer.
And that's what you see in this?
Yes.
And these are all ways that we've tried them in our apiary.
We've mostly used the oxalic acid drip, which is like the syringe.
Some disadvantages: does not target the mites inside the caproot cells where they're reproducing. And its efficacy is reduced when the brood is present. Thymol, another treatment. It's an essential oil from the thyme plant. Treatment takes about 2 to 4 weeks after application. Worker bees detect the gel in this little packet up here, right? Mm-hmm. And then they remove it and move it through the colony. As they clean it off of themselves, it gets stuck to them, and that kind of spreads it around, having an impact on the mites. It's a novel mode of action. So if we're thinking about resistance, it can kind of break up. It can be an alternative to amitraz. Disadvantages: it disrupts the queen egg laying. So there's no clear silver bullet with any of these. And unfortunately, as we see resistance occurring and most likely going to spread, these alternatives may have to be used. And then there's a question of when to apply them. And I asked Jarod to produce this timeline. Maybe you could say a little bit about this.
For a lot of mite treatments, one of the most important factors, but there's 2 important things to keep in mind. It's temperature. If temperatures get too hot, sometimes the products will work too fast and they can start affecting adult bees. And the other thing is honey harvest. So a lot of these products will be less effective if you have a bunch of extra space in the colony during honey production. So you want these products to be in the brood nest where the mites are going to go. So the mites are going to stay in the brood nest. So a lot of these products, I made this timeline using the guidelines the producers made, and I've set it up in a way where
you say the
manufacturers of the product, and I put them here based on what months they would be the most effective.
Okay. So this leads to kind of the next chapter of our story, which is the recent release, approval by the EPA for a new mode of action for varroa mites through the technology of RNAi. So RNAi is interference RNA. It's a way in which the application of double-stranded RNA can be applied to a target organism, in this case, the varroa mite, and selectively shut down the expression of a gene or genes. So as somebody noted at the top of the session, all 3 of these pests for whom resistance has become an issue, a manufacturer has developed an RNAi-based approach for management. So again, RNAi interference RNA, it's delivered through double-stranded RNA that's either fed on or absorbed by the target. But what's interesting about this is the mode of action is very selective in terms of the target gene that's being shut down. So this technology was discovered back in 1998 by Andrew Fire and Craig Mello, who received the Nobel Prize for this as they were working with C. elegans, a nematode. Soon after that, the utility of this to manage the corn rootworm, several Lepidoptera, has been explored. And then in 2022, SmartStack Pro was released that the plant expresses double-stranded RNA targeting the corn rootworm. And then in 2024, we saw the release of Calantha, which is a spray version of this targeting the Colorado potato beetle. Both of these insects have a long history of becoming resistant to insecticides. The corn rootworm, a little while ago, became resistant to BT, which is produced by the corn plant. So an additional mode of action was needed to prevent corn rootworm problems in corn. Colorado potato beetle has a long list of insecticides that it has become resistant to. And so the opportunity to use double-stranded RNA was pretty exciting. What's interesting about RNAi is, unlike, say, a pyrethroid or, say, I don't know, amitraz, the mode of action varies depending upon the form in which that double-stranded RNA takes place, because the manufacturer is targeting a certain part of the insect's biology. So this has been really clearly demonstrated for, say, mosquitoes, where they've explored how you could use double-stranded RNA that attacks the exoskeleton, the chitin cinnate, muscles that affect flight, and then even components of the insect's reproduction, basically becoming like a birth control pill that prevents the insects from reproducing. So even though we're using RNAi here, depending upon what type of double-stranded RNA is used, the mode of action can vary. And this becomes really interesting because we can start thinking about using RNAi to help combat resistance, both in terms of the resistance to the existing insecticides, but maybe in the future, existence to that RNA that's currently out. All right, so this is just a reminder of the current technologies, products that are made available that use RNAi. This is Spark Stacked Pro, or Seed, depending upon the manufacturer. This is where the plant itself is producing RNAi, or the double-stranded RNA. As I mentioned, Calantha, this is an RNAi-based product for Colorado potato beetle. It's sprayed on the plants. And then the most recent one is, and I hope I'm pronouncing this right, Varroa, kind of a clever play on words for an RNAi-based product that's targeting Varroa. And the active ingredient in this is a set of double-stranded RNA that the manufacturer has labeled Vadiscana. Am I pronouncing that?
Vadiscana.
Vadiscana. Thank you, Jared. So what is it? Unfortunately for us in Iowa, we didn't get the EPA approval for use of this product until this spring. So we don't have any data to share in terms of efficacy, but what we are going to do is walk through how we're using it this year. This is not to promote Varroa or the product, but just to show how this would work if you were to use it. Because unlike some of the other mite treatments, the application is going to be really, really critical in terms of its efficacy. So Varroa contains this active ingredient, the double-stranded RNA that's labeled
Vadiscana.
Thank you. The advantages here is, again, it's very specific to the Varroa mites. The double-stranded RNA has been selected to target key components of their growth and development, in this case, their reproductive system. It can affect mites inside the cap cells, getting to them where they're doing most of their damage. It's stable at colony temperatures. Now, the temperature in the colonies can vary quite a bit, but bees regulate that temperature, and within that range, it appears that this product is stable to date. And it's easy to apply. And I put in initials here, Jarod Perez, because Jared just got done using this at our bees in our apiary, and these are pictures that you took while you were applying this. What are some disadvantages? Well, it doesn't directly kill the Varroa mite. It is targeting the reproductive system. So it takes some time for that, say, birth control pill to move through the system. Well, one, move through the colony, become exposed to all the mites, and then that impact being seen in the next generation. The manufacturer claims that it's most effective when mite levels are low. So it's not something that's going to be used when you have a huge population. And it works best with open brood so that the workers who are feeding on the dsRNA infused sugar solution can move it through the colony into those infected brood cells. So how is it applied? Well, what is this, Jared?
This is what the Varroa product comes in. It comes in this little pouch, and there's 3 steps to apply it, and they have the steps labeled there. And I have made some video. I took a video of me applying it to one of our colonies. So the first step is you pull the sticker back. It's actually on there pretty snug. And then after you do that, you want to press down on the top of the packet and it allows the sugar solution to begin dripping out. And then the workers can smell the sugar, and then they'll uptake the sugar, and they'll store it inside the brood food.
Okay, so tell us where you're applying this packet.
So I put this packet on our bottom boards since we were doing some other colony maintenance, but you can apply the packet either somewhere stable that has a flat bottom and where the brood will be.
All right, so on top of this bottom board are going to be the deeps that house the frames in which the brood are maintained.
Yes.
Okay. This is step 2. What's going on here?
So this is where we press down to get the sugar solution to come out of the holes on the packet. And once it starts flowing, the bees will detect that once the box is put together, and they'll start uptaking that sugar solution.
And then this last video is zooming in. You saw
the sugar solution and then me putting the brood nest back on top of the bottom board.
Okay.
So it's a pretty, compared to other treatments, it's very fast to apply.
And so in terms of timing, you applied this Varroa.
I applied it after we did our spring mite treatments. So since you want the mite levels to be low, but when you use this in your mite rotation strategy, you apply your spring treatment first, and then after the spring treatment, you apply the Varroa, and then you do it. We are planning on doing it again in the fall after our honey harvest, our fall mite treatment, then another round of Varroa.
And we'll continue to monitor mites as we
Yes.
have for the last several years, doing mite checks and seeing sort of how well this works this year. But this is our anticipated timeline if mites continue to be a problem.
Yes.
Right. Okay. Okay. Going to be a little bit respectful of time. We've already gone over the 20 minutes. I apologize for that. I'll leave you with a bunch of references here. I believe this gets made available, so maybe you can take a screenshot or otherwise revisit the slide in the webinar later to find all of these. If you need more information about, say, Varroa, the mite itself, the different options, the different management options that we've used, and then there's a couple papers here at the end. The most recent one that came out tying viruses and vectors to colony bee loss, and that has some information about amitraz resistance, and a recent kind of review article about RNAi-based biopesticides, specifically for controlling vector-borne diseases, but consistent with the technology we're seeing here today. Hopefully there's some time for questions. We'll try to answer whatever we can.
Sure, plenty of time, and thank you so much. So we've already got questions coming in, and again, if you have any questions, please type them in that Q&A portion on the bottom of the slide, and I'm just going to start from the top and reading them down and let you answer them. The first question is, would you say that the recent introduction of Amiflex and AM 2.0 is an attempt to reformulate anitraz to slow down the resistance issue with Varroa?
Yeah, I don't know if I can answer that. That's not an issue that we've been exploring. What do you think, Jarod?
It's not something I would know that much about. Sorry.
Okay. Can Varroa be used with honey supers on?
It is not recommended, since you want the honeybees that are taking up the sugar solution to store it in the brood nest. So they recommend applying before and after your honey supers are on and pulled off. If the supers are on, the bees might move the dsRNA into your honey supers. And while
it's not harmful to humans,
it's not harmful to humans, but it won't do anything to help with the Varroa mite problem.
Basically, it would dilute the impact of the double-stranded RNA on the mites, because they'll take that sugar and identify. I don't want to presume the mind of a honeybee, but it's going to take that sugar and take it where sugar goes, which is to the honey cells to be developed into honey, as opposed to going directly to the brood to feed them.
Okay, where would you install if using screen bottom boards with slotted racks above?
So if the bottom board is in a spot where you can put the Varroa packets, you can put it in between. If you have two deeps, put it in between there, or you can put like a shim or a spacer, so that way there's enough space for the bees to crawl on top of the packs to access the sugar solution.
But you wouldn't put this on top of a super.
No.
Yeah, you'd want it to be as close to where the brood are.
Yeah.
To make sure that either the foraging workers and the nursing worker bees get that to the brood as soon as possible.
Yes, you would want the product to be stored as close to the brood nest as possible, or used near the brood nest.
Okay. Can this be used for small or weak hives?
Since the dsRNA is specifically targeting Varroa, it can be used in all size hives. They do recommend to try to not. It's best applied when the colonies are smaller, since it'll get more direct contact with the Varroa mites. If colonies get to really high populations, there'll be more brood cells and they'll be more diluted.
So sorry to interrupt, Laura, but I noticed that a couple of self-identified reps for Varroa are on the call. So there's, like, again, this is not a product endorsement, but I want to make sure that, we get the messaging right and maybe direct more product-specific questions to the reps, like the one who self-identified, Adam Packel. I hope I said that name correctly.
Very good. So we'll continue with the question. So are you going to apply Varroa when there isn't a nectar flow?
That would be the best time to apply it, since there'd be less nectar for the bees to move around and store. It would work the best then.
Yeah, I would say that hopefully it came out clearly that this is our first time using this product, and I think for most of North Americans, this will be their first time. So there's going to be a bit of a learning curve, and part of the reason why we elected to share this at the start of the learning curve is I was a bit surprised that so few of the beekeepers that I work with were aware of this product, and especially the issue that is really at the heart, I think, of its potential, which is to help manage the resistance to amitraz, and so going in the same way that RNAi has been used in other pest systems, corn rootworm, Colorado potato beetle, where they're really intractable pests because of their development of resistance to oftentimes multiple pesticides. We're kind of at the forefront of that with this beast, with the Varroa mite. So, and since it's the #1 pest and really challenging to manage, hopefully that came across with the timeline that we produced. Yeah, there's, even though we're at the start of the learning curve, there could be some big benefits to including this into a management program.
Next question is, approximately how long does uptake take?
I believe on the manufacturer's box, they say depending on how you use it, can take up to 3 weeks. What we notice at our apiary is, even with the nectar flow that's happening right now in Iowa, out of 12 colonies, 11 fully used all the 2 packets of it.
How soon did you see that they had used the packet?
About 3 or 4 days.
Oh, wow.
So they uptook the product really fast for us. And the colony that didn't uptake all of it was smaller, so it was about halfway used. So it took them like about a week. So I would say, at least with my first time using it, it took about a week for the bees to uptake the packets of Varroa. And since our colonies are 2 deeps tall, we had 2 packets, so about 1 liter of sugar syrup solution.
Okay, so maybe this kind of answers, because the next question is, how soon after treatment can regular feeding resume?
Regular feeding, I'm not sure if.
Yeah, that's not a question I think we can ask, because we're not feeding our bees at this point. What we're doing is, let me take a step back. So some beekeepers will artificially feed their colonies with sugar syrup, fondant, at times of low forage availability, maybe through the winter. You applied this a week ago, is that right? So
yeah, about like 10 days.
Yeah, so it's kind of go time for our honeybees right now. We've got quite a bit of dandelion. Now we're starting to see clover. Some of our natives plants are coming online. So this isn't a time when we would artificially feed our bees.
Okay. Do you know what the target gene for Varroa is?
I would know the exact
I
gene.
believe that's a proprietary uh, secret, but the manufacturers made it clear that it's targeting the reproductive system, and I believe it's targeting juvenile hormone in that in its impact on reproduction. So it doesn't immediately kill the mites, but it breaks up that life cycle so that they're unable to reproduce.
Okay.
And
So,
in this way, it's not unlike insect growth regulators that are used in a bunch of different systems. And you have the same kind of relative delay where the insect isn't immediately killed by the insect growth regulator, but some part of their development, either the metamorphosis from immature stage to adult stage is disrupted, or reproduction is disrupted. And then the population, you see a population decline after that.
okay. And I just want to let everyone know that the slides or the recording, not the slides, the recording will be made available and transcribed, and you'll get an email with that information. The next,
Can I pick one? I was looking at I'm curious what Jarod has to say about this. Sometimes hives don't want to take sugar water when you give it to them. Are there any additional stimulants to encourage bees to take the Noroa? Or how about just stimulants to have them take an artificial feeding?
I think the best way in that I have heard is if you want to almost let me try to think about how I should say this. If there's usually they will not like artificial feeding if there's natural forage available for them. So if the concern is that you're afraid, or if the concern is that you don't want to apply something to the colony that they won't use, it would be the best timing to do it would try to put them on when there's not an active nectar flow. And it should increase the odds that they'll uptake the sugar solution.
So that's really going to depend upon the sort of growing conditions, ecology of where
Yeah,
you
the
keep
the
your
timing
bees.
of where they are. So it's hard to say like a specific month or a specific set of weeks because it can change between the years and depending on what region you are. It could also change like when the flowers will begin blooming. Um, but if the question is more of how do I have bees take artificial feed, I don't really have a good answer for that. And besides, if there's stronger forage available in the environment, they prefer that than sugar water.
okay. I'll have to back up to what adjuvant therapy works best after treatment. Adjunctive, sorry. Assuming after 18 weeks in parentheses.
That's yet to be determined. We're really into our very first growing season with this product. So it's, yeah, long-term use of this product within the timeline that, most beekeepers use for mite management. Yeah, I don't think we can say. And what we've heard, and I think I agree with this is not going to be a standalone management practice. It For a variety of reasons. We're still working out sort of the timing of this and how the product is moved through the colony. There's still and as they've indicated, it's not recommended for colonies that have a really large population of mites. So this is not, again, recommended as a standalone, but going to have to be used as a complement to the existing tools that are available to our beekeepers. Yeah, I don't know if there's anything more to say about
I
that.
think, yeah, since we haven't used it and I don't have a lot of experience with it yet, if I had to make an educated guess, if it was like for this treatment we did right now, if I knew need to do another mite treatment, it would most likely be oxalic acid since I can apply that as a drip and keep it out of any honey supers you might have on the colonies. But we haven't done our mite checks for June yet, so I don't know where the mite levels are exactly. Where they were before we applied the treatment was they were all below the treatment threshold. So I'm just curious to see if after using Aurora, if the our mite levels that we see this June, how they compare with last June, because last June they were above our treatment threshold after our spring treatment. So
And-
I don't have data yet,
Yeah,
but
and to one of the participants' question, yeah, we would be interested in some anecdotal feedback. We're going to monitor these colonies like we do all of ours, and we're scheduling, for a treatment in after we harvest. So after late August, when we start preparing the colonies for winter. So yeah, it would be worthwhile hearing from others to get some feedback. We'll share this information with our beekeeping group, and maybe you could have us back on, say a year from now when we've got a better sense of how things worked, not only from our colonies, but regionally.
And I'm going to combine a couple of questions because we had a question about no other treatment should be used for about 18 weeks, yes or no? And then the other question was about using Varroa and Varroa Roxanne at the same time, yes or no?
my understanding from the label of the product is it's best to treat before you add an Aurora. I don't know. I don't know. I haven't tried using it at the same time as other mite treatments, but since its purpose is to control mite reproduction, you want to knock down the mite population, and then you add an Aurora to help keep that mite population low for longer periods of time.
Did you do that with our colonies? Did you
Yes.
apply? Yeah.
We applied formic acid
Yeah.
at the beginning of May, and then about 10 days ago, we applied an Aurora once we confirmed that the treatment was finished. Once we fully finished the formic acid treatment, made sure that the mite resampled for our mite levels to see where they were at, and we added an Aurora.
What were the mite levels like before you did the first treatment?
So before the formic acid treatment, we sampled 5 out of 20 colonies. We had one colony that was about at 8 mites out of 300, so it was very close to the treatment threshold. So we treated all the colonies, and then after the formic acid, it dropped down to about 3. Our highest we found was like 3 mites out of 300. So the formic acid treatment appeared to work, and so now we've applied the Aurora, and so we'll be taking another mite sample at the beginning of June to see where if the mite levels have gone up or changed at all.
And that's part of our regularly scheduled
Yes.
management that we do these.
We sample
assessments
mites
in June.
every month.
Yeah.
So you don't really know yet, but it seems like treating before with a mite something and then following up with the Varroa rather than the Varroa followed by a different treatment.
Yes.
Yeah, because the issue there is, as the manufacturers noted, it's not for treating heavily infested colonies.
Varroa is kind of like a follow-up treatment as part of like the plan. It's like your second step after your initial treatment. It's like the next step you take to try to keep that initial treatment as effective for as long as possible, to kind of like extend the effects of that first treatment.
Yeah. Next question, better to apply Varroa during a dearth to maximize efficacy?
Yes. If there's a foraging dearth, then there will be less competition between where the foraging bees will go to for sugar syrup for nectar versus the sugar syrup. So it'll make the sugar syrup more attractive and they'll uptake it faster.
Yeah. Does it have a shelf life or expiration date?
Yeah, that's a good question. So my general understanding with anything like with RNA, DNA, the issue is photodegradation when you apply these in the field. So this is in part why this is kind of a nice setting. Those sackets, they're in the colony, they're not going to be exposed to sunlight. I think they're fair. What I've heard from the manufacturers, they're fairly shelf stable. This isn't something that needs to be used immediately. And again, maybe one of the product reps could comment on that.
The next question was, my understanding is that both formic and oxalic as certified organic treatments are not expected to develop resistance. Would someone still need to use Varroa in rotation with the acids?
Oh, that's a good question. So I'm not sure I could say that any form of management is resistance proof. If it's overly relied upon. I think one of the things we've seen with formic acid, oxalic, is there are pros and cons. It doesn't always get to where the mites are in a way that this technology may be able to. Again, I think we don't know yet. We're in the early stages of using this, but there's the potential for this to complement the existing management tools, so that you can stay on top of the population and keep them from exploding and becoming what some of the biologists call mite bombs, where you just have so many mites in a colony that they end up, spreading out to the other colonies in the apiary. What do you think?
With my experience using formic, thymol, oxalic acid, is they work, but we have had cases where we did that treatment to colonies that had mites, and the mite levels don't go down. We've even had some colonies where the levels go up post treatment. So while it might not, I I don't know how. I wouldn't say they're like foolproof, or like if you apply them, they'll work 100% of the time. And I also don't believe they would be fully mites would never be able to develop resistance as well. I think it's yeah, so I'm tripping over my words, but
Yeah, we have the larger issue here where it's new technology, and we still have a lot to learn about how best to use it effectively, efficiently, and then sort of sustainably. Because, like I said, any pest management tool, if it's overly relied upon, there's a potential for resistance to develop to it. And gosh, if this tool is as effective as we think it could be, I'd hate to see it lost to us in a short period of time.
We had a question.
Sorry.
Sorry.
I was just gonna say, and that kind of, I'm looking through some of the questions. Are there any other RNAi developments? Yeah, I can't speak to that for this manufacturer, this product, but the general topic of using RNA interference and double-stranded RNA to generate that is a very active area of research because of the potential targets that are available to us when we use that technology. The challenge has been getting the double-stranded RNA into the target organism. And so sometimes there are pests where if you spray them, they'll either ingest it or it'll be absorbed. There's a couple of cases of that for some of the insects that I work on, like this, like aphids. But oftentimes it has to be ingested. In the case of the corn rootworm, they have to eat the plant tissue that is expressing the double-stranded RNA. And in this case, it's really kind of clever. The honeybees are they're feeding on that sugar solution that has a double-stranded RNA in it. And then the mites are feeding on those bees to get you the...
They're also mixing the double-stranded RNA into the brood food. So the brood they're feeding, it uptakes the double-stranded RNA as well. And then when the mites enter the cell and feed on them, that's how so it also targets the mites that aren't outside the cells as well. So it works, or it should work for mites inside the cell and mites outside the cell. I think its main target is the mites that go into the cell where the brood and feed on them.
Okay, kind of skim through. So is it, if there's portions of it that are unconsumed, can it be saved at all, or is it kind of stuck?
Most likely not.
How long did it take for even your slow, small colony to...
I believe the most slow, small colonies took about 8 or 9 days. And it just depends on like what's available in the environment, and also that colony was smaller, so they had less workers to move the sugar solution into the colony. So it's kind of hard to say. I would say generally best practice is if you put a treatment on a colony and there's some left over, not to try to save any of it, because I don't think it would be. I won't speak about how stable that would be, but I wouldn't try to reuse any. If you take the packet off after like 3 weeks and there's still sugar solution in there, I wouldn't try to save that. I don't think it would be very effective if you tried to reuse it.
So there was a question about costs. Yeah, how much did this cost?
I'm trying to think of the number. We bought enough treatment for 24 colonies. And I think it was at least $400. I don't remember. We had problems.
Wow.
It's for right now it's a kind of costly
Mhm.
compared to other treatments.
So when you purchased a box and that box came with two packets, right?
Yes.
And then it's two packets per colony. So you're buying treatment for just one
it's
colony.
one packet per brood box, and all of our colonies are two
Oh,
brood
right.
boxes, so we needed two packets for each
Yeah,
colony.
right. Okay.
Um,
Okay, I'm kind of skipping around. Is this labeled organic?
That I don't know.
I'm not sure.
Yeah, so RNAi, I believe for uh, the Colorado potato beetle is listed as a biopesticide. I'm not, and I don't know. I shouldn't speculate if that's approved for organic use or not.
Mhm. This kind of interesting question. Does a rapid degradation of dsRNA by honeybee enzymes in the hive pose a greater technical challenge for commercial RNAi development than the potential off-target effects on other arthropods like monarch butterflies?
I got to read through that question. That was does the rapid degradation of double-stranded honeybees in the hive pose a greater technical challenge?
Yeah, I'm not.
So, oh gosh, that's a really interesting, but also kind of challenging question to answer because um, the way in which a herbivore feeds on plant tissue is a little bit different than the way honeybees feed and use um, sugar or nectar. Um, so the worker bees that would feed on the sugar solution, feed is I'm gonna sorry, I'm gonna use air quotes. They're not actually feeding on that nectar. They're holding it in a honey crop, and then that gets passed on to other bees or directly to the brood. So it's not to say that there isn't some enzymatic activity going on there, but it's not to the full degree of something like a corn rootworm that's feeding on tissue and that's moving all the way through their gut and being processed for nutritional value. Does that make sense? It's yeah, I'm not quite sure I can, but I don't quite know if I can answer that with regard to where the questioner was going with that, with say something like the monarch butterfly. It's I think the challenge with RNAi as a platform, a technology for pest management is it's really on a case by case basis, depending upon how the product is applied, the mode of action, and of course the target pest and where that pest is being targeted.
Okay, I'm gonna combine a couple questions here. So does it work on nymph or adult Varroa or both? How long does it stay effective? And you're evaluating the effectiveness now, right?
Yes, we just finished the application, so I don't have any data yet on how effective it could be. But for if it targets both, I don't know exactly how it interacts with the adult mites. That would be some of the people in the chat that are from the company, but I believe its primary target is the nymphs while they're developing before they're able to actually lay any viable eggs.
Okay.
And then
And
I was just gonna, sorry, I had a question for Jarod. So you've gone out and inspected the colonies. You've seen that you removed the packaging.
Yes.
Then after that, okay.
For the packages, I had them on the bottom board, so I just took the colonies apart, took them off the bottom board, and then put them back on with gloves.
Okay. That answers the what procedure you used to remove the packaging. Okay, I don't see any other questions. I just want to remind everyone to read your product label. We've done our best to answer those questions about how to apply it and things like that, but you certainly want to read that label and certainly get in touch with the manufacturer with any detailed questions. So thank you both so much. I think that was a record number of questions that we've had, and there's certainly a lot of interest in it and good attendance. Before I let everyone go, I want to let you know that our next Pest in Progress webinar will be on Wednesday, July 15th at 12 p.m. Central, 1 p.m. Eastern. Ashley St. Clair from Corteva AgriScience, she's a regulatory ecotoxicologist, will be talking again about honeybees, looking at AI-driven image analysis for assessing honeybee colony health in regulatory fields. So we will continue our honeybee health theme for the summer.
Yeah, and hey, thank you very much for giving us the opportunity to share what little we know about this new product. Hopefully, it's of value to your audience. And again, appreciate that. There are other people out there that have a little bit more experience with this. Of course, a manufacturer, and to just echo what you said, label is the law.
Exactly. So thank you both so much. Thank you, everyone, for attending, and we will look forward to an update in about a year.
Thanks.
Thank you.
Bye. Do we?